Welcome back Bio Squad. This is episode 3 of the Break It Down series, and today's episode will be dealing with enzymes. I got you all covered with various types of questions, and here is the place to start quality revision. If you are new, join the squad right now, as all we do is keep growing. Hit that subscribe button and do leave a like too. Let's get this revision started. Question 1. A fixed volume of the enzyme catalase was added to a fixed volume of hydrogen peroxide solution. The diagram shows how the rate of the reaction changed over the course of the reaction. Why did the actual rate of reaction decrease over time? The keywords over here are a fixed volume of the enzyme, a fixed volume of hydrogen peroxide, which is the substrate. If there is a fixed volume of substrate added, that is of hydrogen peroxide, then over time the substrate molecules will get used up, or the substrate concentration decreases over time. Therefore, if we go to the answers A, B, C and D, the best possible answer is D. The substrate molecules were used up. Why cannot be either A, B or C? Well, if you look at A, the enzyme active sites become saturated. The enzyme active sites become saturated initially, not over time. Therefore, A is incorrect. B, the enzymes were denatured. There is no external factor that causes the enzymes to be denatured, and according to this reaction and the diagram given, we cannot say that the enzymes were denatured by looking at the actual rate decreasing over time. If they have mentioned changes in temperature or changes in pH or any other external factor, then we could say that B is the correct answer. However, there is no mention of external factors being changed, hence B is not a possible answer. C, the product inhibited the reaction. When the enzyme catalyzes hydrogen peroxide, it will break it down to water and oxygen, and water and oxygen cannot inhibit the reaction. Therefore, answer C is also incorrect. The best possible answer is D. The substrate molecules were used up as the reaction proceeds over time. Question 2. Which levels of protein structure are always involved when competitive and non-competitive inhibitors bind to enzymes? In these kind of questions, you should always look out for the word tertiary structure, because tertiary structure, they form part of the active site and is important to the active site of an enzyme. Therefore, which levels of protein structure are involved when competitive and non-competitive inhibitors bind to enzymes? It should be tertiary structure only, because tertiary structure forms part of the active site and the active site is concerned with the binding of inhibitors. And all enzymes do have a tertiary structure, and tertiary structure is what determines the shape and function of the enzyme. Hence, when competitive and non-competitive inhibitors are involved, tertiary structure is affected. That leaves us with the answer being D. Question 3. The enzyme lysozyme secreted from tear glands forms deposits on contact lenses. Which ingredient would be effective in a contact lens cleaner for removing these deposits? A. Ethanol. B. Lysosomes. C. pH buffers. D. Proteases. Key thing you should remember is that enzymes are proteins. Hence, they would be broken down by enzymes known as proteases, as proteases, as the name suggests, breaks down proteins. Therefore, in order to remove the deposits forming on contact lenses, you would need an ingredient which could digest or break down the deposits, leaving us with the best possible answer, that is D, proteases. Question 4. The graph shows the activation energy of an enzyme catalyzed reaction and the same reaction without a catalyst. Which of the following shows the activation energy of the uncatalyzed reaction? Now, when an enzyme is added, always the activation energy of a particular reaction will be lowered or decreased. However, since they are asking which shows the activation energy of the uncatalyzed reaction, then it should be X plus Y, because when catalyzed the activation energy is lowered, hence for uncatalyzed the activation energy will be higher. And if you look at the peaks, the lower peak represents the one with the enzyme being added, hence the lower activation energy. The higher peak or the upper peak represents the activation energy of the uncatalyzed reaction. And how do you get the activation energy of the uncatalyzed reaction? It is by adding X and Y. Therefore, X plus Y will give you the activation energy of the uncatalyzed reaction, leaving us with the answer being C. Question 5. Which of the following statements are true of all enzymes? 1. Soluble in water. 2. Catalyze the breakdown of large molecules into smaller molecules. 3. Only have one active site. 4. Have a quaternary structure. All enzymes do not break down molecules. Some enzymes can even build up larger molecules from smaller ones. Hence, answers having 2 in it are incorrect, leaving us with the answer being either C or D. And not all enzymes have a quaternary structure, but all enzymes do have a tertiary structure. Therefore, 4 is incorrect. It is not true of all enzymes. The keyword over here is all enzymes. And one thing you should know is that all enzymes are actually soluble in water, leaving us with the answer being C, 1 only. Question 6. The graph shows the effect of temperature and pH on enzyme activity. Which statement explains the enzyme activity at the points shown? A. At P, hydrogen bonds are formed between enzyme and substrate. B. At Q, the kinetic energy of enzyme and substrate is highest. C. At R, peptide bonds in the enzyme begin to break. D. At S, the enzyme is completely denatured. Statement D is obviously incorrect because pH doesn't lead to enzyme denaturation, only temperature does. pH just changes the overall tertiary structure and affects the function. It doesn't lead to complete denaturation. Therefore, D is incorrect. C. At R, peptide bonds in the enzyme begin to break. R represents the rate of reaction according to the change in pH, and pH doesn't affect peptide bonds because peptide bonds are only located in the primary structure, and primary structure doesn't change or is unaffected by the pH. Therefore, C is also incorrect. B. At Q, the kinetic energy of enzyme and substrate is highest. If you look at Q, which is a point marked on the graph of the rate of reaction plotted against temperature, kinetic energy will be highest at the highest temperature, not at Q. Therefore, statement B is also incorrect, leaving us with the only possible answer, that is A. At P, hydrogen bonds are formed between enzyme and substrate. That is a possible answer because when a substrate binds with an enzyme, temporary bonds hold the two molecules together. Therefore, A is a possible statement. Question 7. In an enzyme catalyzed reaction, which combination of inhibitor and substrate would result in the highest rate of reaction? Key thing you should know is that non-competitive inhibitors leads to no or slowest reaction. Therefore, answers having non-competitive inhibitors are incorrect, leaving us with either A or B. When substrate concentration is highest for competitive, then the rate of reaction is highest. And since the question asks which would result in the highest rate of reaction, the answer is A. Inhibitor is competitive, substrate concentration should be high, because if the inhibitor concentration is higher than the substrate concentration, then the rate of reaction will be less. Hence, in order to result in the highest rate of reaction, the substrate concentration should be higher than the inhibitor concentration. Then only the reaction would occur. Therefore, A is the correct answer. Question 8. Which graph represents the changes in concentration of a substrate and its product in the same enzyme catalyzed reaction? Over time, the concentration of product increases and substrate decreases because in an enzyme catalyzed reaction, you provide the substrate to the enzyme, and then when the substrate binds with the enzyme, product would be formed. Hence, the product concentration increases, and it doesn't increase in a linear form. It increases gradually, hence is represented by a curve, not a linear graph. Therefore, answers C and D are incorrect. And we should look at the key. The continuous line represents the product, and since the concentration of product increases over time, the answer should be A. Question 9. Glucose in urine can be detected using a biochemical test. When the end of a test strip, which is impregnated with the enzyme glucose oxidase, is dipped into urine, the development of a blue color indicates that glucose is present. This is a reliable test that people with diabetes can carry out at home. Which feature of the enzyme makes this test so reliable? Enzymes are specific in nature and they only work on a specific substrate. And that is what makes each enzyme unique, because each enzyme is specific in nature and it only works on a specific substrate. It doesn't work on any other substrates. If you go to the answers A, B, C and D, the best possible answer is B. It is specific. Why are the other answers incorrect? Because the other features do apply to other enzymes as well, not only to this specific one. In order to make this test as reliable as possible, the enzyme should be specific because it works on a certain substrate only. It doesn't work on any other substrates. Therefore, B is the correct choice. Question 10. What is the role of enzymes in metabolism? A. To catalyze the hydrolysis of large molecules only. B. To increase the number of collisions between molecules. C. To lower the activation energy required to start a reaction. D. To supply the activation energy required to start a reaction. Key thing to note is that enzymes are used generally to lower the activation energy of a reaction by providing an alternative pathway. Therefore, if we go to the answers A, B, C and D, the best possible answer is C. To lower the activation energy required to start a reaction. Why are the other answers incorrect? It is because if you look at A, to catalyze the hydrolysis of large molecules only, that is incorrect because enzymes can be used for breakdown as well as for build-up of large molecules. Therefore, A is incorrect. B. To increase the number of collisions between molecules. That is not the main role of enzymes. The main role of enzymes is to lower the activation energy in order to provide a faster rate of reaction. Hence, B is also incorrect. D. To supply the activation energy required to start reaction. Enzymes do not supply any energy. They just lower the activation energy, or in other words, they cause a change in the activation energy. No energy is supplied. Therefore, D is incorrect, leaving us with the only possible answer, that is C. To lower the activation energy required to start a reaction. Question 11. Which is correct for a non-competitive inhibitor of enzyme action? 1. Increasing the concentration of the enzyme substrate will reduce its effect. 2. It reduces the activation energy required for a reaction to take place. 3. It reduces the maximum rate of reaction. If you look at statement 1, increasing the concentration of the enzyme substrate has nothing to do with the effect because non-competitive inhibition usually leads to reduced or no rate of reaction. Therefore, statement 1 is incorrect. Let's look at statement 2. It reduces the activation energy. Again, inhibitors have nothing to do with activation energy. 3. It reduces the maximum rate of reaction. Yes, that is correct. Over here, maximum rate of reaction refers to Vmax. And if you recall how Vmax changes when in the presence of an inhibitor, if it is a competitive inhibitor, Vmax is unchanged or unaffected. If it is a non-competitive inhibitor, the Vmax is reduced. That is, the maximum rate of reaction is reduced, leaving us with the best possible answer, that is B, 3 only. Question 12. Which words from the table correctly complete the paragraph about enzymes? When the pH of an environment is decreased below an enzyme's optimum pH, 1 bonds between adjacent 2 groups holding the 3 structure are disrupted. pH normally affects the ionic bonds and it doesn't affect peptide bonds because peptide bonds are within the primary structure and pH has nothing to do with the primary structure. Hence, answers having peptide in it are incorrect, leaving us with the answer being either A or B. And again, pH affects ionic bonds. Hence, the best possible answer is A. Hydrogen and ionic bonds between adjacent R groups holding the tertiary structure are disrupted. Yes, because tertiary structures, they form the main part of the enzyme, that is the active site, and also determines the shape and function of the enzyme. Hence, if pH changes, it affects the overall shape of the enzyme as well as tertiary structure. Therefore, A is the correct answer. Question 13. Which levels of protein structure are always involved when competitive and non-competitive inhibitors bind to enzymes? This is the same question as done previously, although it is from a different paper. Refer to question 2 for the explanation. Let's move on to the next one. Question 14. Which statements are true about the optimum temperature of all enzymes? 1. It is the temperature at which enzymes work best. 2. It is the highest temperature at which the enzyme will work. 3. It is between 35 degrees Celsius and 40 degrees Celsius. Over here, the keyword is all enzymes. And if you recall the definition of optimum temperature, it is the temperature at which the enzymes work best. Therefore, statement 1 should be true. Let's look at statement 2 and 3. It is the highest temperature at which the enzyme will work. That is incorrect because some enzymes work at low temperatures and some enzymes work at very high temperatures. Therefore, it is not true for all enzymes. 3. It is between 35 and 40 degrees Celsius. That is also incorrect. Again, some enzymes could work at very low temperatures. Some enzymes could work at above 40 degrees Celsius. Therefore, it is not true for all enzymes, leaving us with the best possible answer, that is C, 1 only. Question 15. The rate of enzyme catalyzed reactions in human cells is regulated. What may be involved in such regulation? 1. A change in enzyme concentration. 2. A change in substrate concentration. 3. Inhibition by the final product of the reaction. Enzyme catalyzed reactions are normally regulated by enzyme concentration, substrate concentration as well as inhibitor. Therefore, all three factors are involved in such regulations. Hence, the answer is A, 1, 2 and 3. Question 16. Which statement correctly describes the action of comparative enzyme inhibitors? Comparative inhibitors are those that bind to the active site of the enzyme, and the action is that they limit the number of enzyme substrate complexes. Therefore, if you go to the answers A, B, C and D, the answer is C. They limit the formation of enzyme substrate complexes. Why are the other answers incorrect? A. They bind permanently to the active site. They do not bind permanently. As comparative inhibitors, they are generally of reversible inhibition. Hence, do not bind permanently. Hence, A is incorrect. B. They change the shape of the active site. That is also incorrect because only non-comparative inhibitors change the shape of the active site by binding to a site other than the active site. Hence, B is incorrect. D. They lower the activation energy. That is not true because inhibitors have nothing to do with activation energy. Only enzymes are able to lower the activation energy of a reaction. Therefore, D is also incorrect. Question 17. Two enzymes X and Y were used in an experiment. Enzyme X was from bacteria that live in rivers and lakes at temperatures from 5 degrees Celsius to 20 degrees Celsius. Enzyme Y was from bacteria that live in hot water springs at temperatures from 40 degrees Celsius to 85 degrees Celsius. The experiment measured the concentration of product produced by each enzyme at temperatures between 0 and 100 degrees Celsius after 5 minutes. Which graph shows the results? Keywords over here are concentration of product produced upon temperature. Therefore, since they are asking which graph shows the results, enzyme X works at relatively lower temperature than enzyme Y, as enzyme Y works at around 40 to 85 degrees Celsius. Therefore, product concentration will be steepest at temperatures towards that range, that is at higher temperature ranges. And if you go to the answers A, B, C and D, answer A is incorrect because for Y at 0 degrees Celsius the concentration of product should be 0, not in between. Therefore, A is incorrect. Let's look at B. Yes, B is the right answer because enzyme Y works at high temperatures, hence product concentration should be highest towards those temperatures, and X works at 5 to 20 degrees Celsius. Therefore, concentration of product should be in that temperature range, and above 20 degrees Celsius there should be 0 concentration of product as the enzyme would be denatured. Why are C and D incorrect? Because concentration of product increases in D no matter the temperature for both X and Y. Therefore, that is incorrect because enzyme X would be denatured at high temperatures. And at C, concentration of product at high temperatures for X remains constant. However, that cannot be the case as the graph plotted is the concentration of product produced, not the concentration of product that is left behind. Since it is of product being produced, X will not produce products at high temperature ranges. Therefore, C is also incorrect, leaving us with the best possible answer, that is B. Question 18. An enzyme was added to a small excess of its substrate. All variables were kept constant. A student was asked to sketch a graph to show how the concentration of the enzyme substrate complex changes over time. Which graph shows this correctly? Initially, there would be a maximum enzyme substrate complexes occurring, after which, over time, it would decrease as the substrate concentration will also decrease. Because they have mentioned small excess of its substrate, it is not an infinite amount of substrate concentration. Hence, initially there would be the highest enzyme substrate complexes being formed, and over time it would decrease, leaving us with the best possible answer, that is D, as only D represents the correct graph. Concentration being highest initially and over time decreasing. Question 19. The following statements are about enzymes. 1. Folding of an enzyme molecule causes the formation of the active site. 2. The shape of the active site changes to enable the substrate to bind. 3. Temporary bonds hold the substrate in the active site. 4. More enzyme substrate complexes are formed at the optimum temperature. Which statements are correct for the induced fit hypothesis? Induced fit is where the enzyme is able to change shape and mold around the substrate to allow better binding. Therefore, if you go to the following statements, statement 1 is incorrect because folding of an enzyme molecule causes the formation of the active site doesn't describe the induced fit. It just describes the general formation of an enzyme molecule, not the induced fit specifically. Therefore, answers having 1 in it are incorrect. Let's look at answers 2, 3 and 4. Well, all the other statements, that is 2, 3 and 4, are correct. Let's look at 2. The shape of the active site changes to enable the substrate to bind. Yes, that does describe the induced fit hypothesis. 3. Temporary bonds hold the substrate in the active site. That again is part of the induced fit, because after it binds with the substrate, then temporary bonds will continue to hold the substrate in the active site. 4. More enzyme substrate complexes are formed at the optimum temperature. That is also true of induced fit hypothesis as well as of the lock and key model. Hence, statement 4 is also correct. Therefore, the answer is C, 2, 3 and 4. Question 20. How is the Michaelis-Menten constant used? Km generally determines the affinity of the enzyme towards a substrate. Therefore, if you go to the answers A, B, C and D, the best possible answer is B, to compare the affinity of enzymes for their substrate. The other answers A, C and D do not talk about the affinity of an enzyme, hence they are incorrect. Question 21. The diagram shows an enzyme, its substrate and an enzyme substrate complex. Which statement explains how the substrate is able to enter the active site of the enzyme? This diagram right here represents the induced fit hypothesis. As I explained in an earlier question, induced fit hypothesis is where the enzyme is able to change shape and mold around the substrate, enabling better binding of the enzyme and substrate, hence forming an enzyme substrate complex. Therefore, if we go to the answers A, B, C and D, the best possible answer is A. Contact between the substrate and the enzyme causes a change in the enzyme shape, as that is clearly visible in this diagram. Let's look at B, C and D. B. The shape of the active site and the shape of the substrate are complementary. That is incorrect, because as you can see, the substrate molecule has a larger size than the enzyme's active site. Hence, it is not complementary in shape. C. The substrate within the active site forms hydrogen bonds with amino acids. That doesn't explain how the substrate is able to enter the active site. That describes the process after what happens when they bind. Therefore, C is incorrect. D. When the enzyme substrate complex forms, the tertiary structure of the enzyme changes. Enzyme substrate complex doesn't lead to change in tertiary structure of an enzyme. Tertiary structure of an enzyme only changes when it is affected by factors such as temperature or pH. And tertiary structure is generally affected when bonds holding the structure change or break. Therefore, D is also incorrect. Since the question refers to how the substrate is able to enter the active site, it should be about the enzyme being able to change in shape to allow a better fit. Hence, A is the correct answer. Question 22. What could be used to calculate the rate of an enzyme catalyzed reaction? 1. The appearance of product. 2. The disappearance of substrate. 3. The Michaelis-Menten constant. The rate of an enzyme catalyzed reaction is generally calculated by the amount of product formed or the amount of substrate used. Hence, the best possible answer is A, 1 and 2 only, because the rate of reaction is generally defined as the amount of product formed over time or the amount of substrate used over time. Question 23. What is the effect of an enzyme in an enzyme catalyzed reaction? Key thing you should know by now is that all enzymes lower the activation energy. Hence, the answers being either A or B decreases the activation energy. Also, if I haven't mentioned it earlier, enzymes has no effect on the energy yield. They just lower the activation energy. They do not contribute to producing energy or taking away energy. Therefore, it has no effect on the energy yield. So you may ask, how does it lower the activation energy without having an effect on the energy yield? Well, enzymes are able to lower the activation energy of a particular reaction by taking an alternative pathway that uses less energy than the current pathway. Hence, that is how it is able to decrease the activation energy without having an effect on the energy yield. Question 24. Which statement is only true for the induced fit theory of enzyme action? Induced fit is again where the enzyme is able to change shape. And if you go to the answers A, B, C and D, the only answer that talks about the enzyme changing shape is answer C. The enzyme changes shape in the presence of the substrate, and that is also the correct answer. Question 25. The effect of substrate concentrations on an enzyme catalyzed reaction was measured in 3 different conditions: with no inhibitor, with inhibitor X, with inhibitor Y. The graph shows the results. Which statement is correct? Key thing you should know is that there are 2 types of inhibitors known as comparative inhibitor and non-comparative inhibitor. When in the presence of a comparative inhibitor, the Vmax or the highest rate of reaction will be unchanged or unaffected. Therefore, inhibitor X represents comparative inhibitor, as it does reach the highest rate of reaction similar to that of enzyme with no inhibitor. Hence, X is of comparative inhibitor. Non-comparative inhibitors, they cause a change in the Vmax or the highest rate of reaction. And how do they cause the change? By lowering the Vmax or the rate of reaction. Hence, inhibitor Y represents non-comparative inhibitor. Let's go through the answers A, B, C and D. A. X is a comparative inhibitor. That is correct, which binds away from the active site. That is incorrect, because comparative inhibitors, they bind to the active site, not away from the active site. B. X is a non-comparative inhibitor. That is incorrect, because X represents comparative inhibitor. C. Y is a comparative inhibitor. That is also incorrect, because Y is a non-comparative inhibitor, as it causes a change in the rate of reaction. D. Y is a non-comparative inhibitor which binds away from the active site of the enzyme. That is correct, because non-comparative inhibitors, they bind away from the active site. They bind to a region that is not part of the active site of an enzyme. Hence, D is the correct answer. Question 26. The graph shows a concentration of one of the substances which is involved in an enzyme catalyzed reaction. Which substance is shown by line X? Over time, enzyme substrate complex and substrate changes. However, enzyme concentration doesn't change unless other external factors such as temperature conditions changes, which leads to enzyme denaturation. However, in this particular reaction, they haven't mentioned any change of temperature. Therefore, the enzyme cannot be denatured. Hence, the enzyme concentration should remain the same throughout. Therefore, line X represents the enzyme concentration, leaving us with the answer being A. Question 27. The graph shows the effect of substrate concentration on the initial rate of an enzyme catalyzed reaction. The enzyme concentration is constant. Which statement about the graph is correct? I have marked over here which one is which. Initially, for a particular rate of reaction, the substrate concentration would be limiting, and towards the end, the enzyme concentration would be limiting as the substrate concentration continues to increase. You have to be very careful on what the particular graph is about. Over here, the x-axis represents the substrate concentration. Hence, towards the end, substrate concentration cannot be limiting, as substrate concentration is increasing as you move from left to right. Therefore, region S to T should represent the enzyme concentration being limiting. Hence, if you look at the answers A, B, C and D, the best possible answer is C. Between S and T, the number of enzyme molecules is limiting the rate of reaction. Why are the other answers incorrect? A. Between R and S, the number of enzyme molecules is limiting. That is incorrect, because enzyme molecule is only limiting between S and T. Between R and S, the number of product molecules is limiting. Product molecules has nothing to do with limiting the rate. Hence, it is incorrect. D. Between S and T, the number of substrate molecules is limiting. That is also incorrect, because substrate concentration is increasing as you proceed. Therefore, towards the end of a reaction, it cannot be limiting. Only towards the start, the substrate concentration could be limiting. Hence, D is incorrect. Question 28. The diagram represents a reversible interaction between the active site of an enzyme and different inhibitors X and Y. Which row correctly identifies the type of inhibition shown by inhibitor X and inhibitor Y? Key thing you should know is that if an inhibitor binds to the active site, it is a comparative inhibitor, and if an inhibitor binds away from an active site, that is to a site that is not part of an active site, then that is a non-comparative inhibitor. Hence, inhibitor X represents comparative inhibitor as it binds to the active site, and inhibitor Y represents non-comparative inhibitor as it doesn't bind to the active site but away from it. Therefore, the answer is B. Question 29. Aspirin inhibits an enzyme by reacting with an amino acid that forms an essential part of the 3D structure of the enzyme. Part of the aspirin molecule binds firmly with the amino acid. What describes this inhibition? 1. Competitive inhibition. 2. Non-competitive inhibition. 3. Reversible inhibition. If it binds to an amino acid instead of the active site, then it is non-competitive inhibition. And non-competitive inhibition is generally not a reversible inhibition. As it says towards the end, part of the aspirin molecule binds firmly with the amino acid. Hence, it cannot be reversible inhibition, leaving us with the only answer, that is D, 2 only. Question 30. The table shows the Michaelis-Menten constant Km for 3 enzymes. Which interpretation of the information is correct? A. Enzyme C has a Vmax which is half that of enzyme P. B. Enzyme C will reach Vmax in the shortest time interval. C. Enzyme F has the greatest affinity for its substrate. D. Enzyme P has a Vmax of 6 into 10 to the power minus 3 millimoles per dm cube. Now, a key thing is that Km generally talks about the affinity of an enzyme towards a substrate. Hence, higher the Km value, lower the affinity, and vice versa. That is, lower the Km value, higher the affinity. They both have an opposite correlation. Again, higher the Km, lower the affinity, lower the Km, higher the affinity. And if you go to the table, enzyme F has the lowest Km value, hence will have the highest affinity out of all the given enzymes. And that is only shown by answer C. Enzyme F has the greatest affinity for its substrate, because they are asking which interpretation of the information is correct. Therefore, we should refer to the information that is given in the table, and from that information we can only conclude that enzyme F will have the greatest affinity. Hence, the answer is C. The other answers talk about Vmax, which we cannot say from the given table information. Therefore, they are incorrect. There we go. 30 questions done and dusted. Please do leave a like, comment, and share with whomever you know. Let's get this bio squad growing. Meanwhile, just a reminder, if you haven't already, subscribe for more content and stay tuned for more.